In the first part of the cell membrane presentation, we talked about the structure of the cell membrane. And now we're going to talk about how the cell membrane controls what gets in and out of your cell. So again, this is just an overview of the structure of the membrane. So there is the phospholipid bilayer, which is the double layer of phospholipids. And then there are proteins inserted in the phospholipid bilayer. So for things like water and glucose and sodium and amino acids, they have to, if they can get into the cell or out of the cell, they either have to get through the phospholipid bilayer or go through one of the protein channels, would be the only two ways of things to get in or out of the cell. So the types of ways that things can get in and out of the cell is what we're going to talk about. Your cell membrane, or remember plasma membrane and cell membrane mean the same thing, is selectively permeable. You can kind of get what that means just from the words, in that selectively permeable means some things can get through across the membrane, other things can't. One of the major determinants is size, so little things can get through easier than big things, but even then there's some selection as to what can go through your cells. There are two primary ways for movement in and out of cells. One is called passive transport, and that's also called diffusion. And passive transport or diffusion does not require energy. It's the way molecules will naturally flow. Molecules will go from where there's more to where there's less. So if you were to hold a bottle of perfume and squirt the perfume, initially there's more molecules of perfume right outside of the atomizer, but they're going to slowly spread out throughout the entire room. That's passive transport or diffusion. So concentration is from where there's more to where less. So molecules will go from where there's more of them to where there's fewer or less of it. Active transport, on the other hand, is movement the opposite way. That does require energy. That's where things go from where there's less to where there's more. So that's not the kind of natural movement of molecules. So you have to expend energy to do that. And we're going to talk about each of these. So before we get into the diffusion, I just wanted to again define what a solution is. So a solution has a liquid, which is called the solvent, and then it has something dissolved in the liquid, which is called the solute. So if you had water and you put sugar in it, water would be the solvent and the sugar would be the solute. So since all of the solutions inside of our body are water-based, we're going to be talking about movement of molecules or solute in water-based solutions. So diffusion is the movement of solute down a concentration gradient from where there's more to where there's less. Things will go in both ways, but the overall change is going to be from where there's more, where they're high, to low. When equilibrium is when the concentration is equal on both sides, and then you get no additional net movement because the concentration is equal on each side. So let me show you some pictures of that. So an example of diffusion. So we have these two goldfish in a fishbowl, and we put a little crystal of green dye in the water. And so initially all the molecules of green dye are together in that crystal, but with time they're going to diffuse away from the crystal until they're equally dispersed throughout the water. So when they're equally dispersed, that's equilibrium. And the process of the molecules of dye moving is called diffusion. They're going from where there's more, which would be the crystal, to where there's less, which is the water, until they're equally distributed. And that's what diffusion is. So diffusion, if you think of it in that fishbowl, we didn't have a membrane, but diffusion would be molecules going through the membrane from where there's more to where there's less. And that's called passive transport or diffusion. Those two mean the same thing. So if we start out with these little green circles, we have many more of them on the outside than on the inside. So providing they can get through the membrane, they're going to go from where there's more of them to where there's less of them. If there was more on the inside, they would go the other way. And that requires no energy. There's some transdermal patches. I don't know if any of you have ever used these, and they have a concentration of some kind of medicine inside the patch, and it slowly diffuses through your skin. These are for motion sickness. There's some birth control patches. There's patches for smoking cessation, but they use the process of diffusion to deliver the medication over time. This is just another picture. So here's your phospholipid bilayer. Now, if the molecule, this one, there's a bunch of these little green things outside of the cell. If it's able to diffuse in lipid or dissolve in lipid, it can go right through the phospholipid bilayer to get inside of the cell. It will do that by diffusion from where there's a high concentration to where there's a low concentration. For water we're going to talk about water in particular now, because water is a special case of diffusion. And the diffusion of water has a special name. It's called osmosis. So it's still diffusion. It's still water going from where there's more water to less water. It changed my slides. It changed slides, so I can't go back to that one. But I think we're okay. We'll just keep going with this one. So osmosis is the diffusion of water from where there's more water to where there's less water. And this gets more complicated because water, if you remember, is a solvent. And so there's stuff dissolved in the water. And so there's more water when there's less stuff dissolved in it. Let me try to explain that a little better. So suppose you have a solution of salt in water, and it's 95% water and 5% salt. And then separated by a membrane, this is supposedly a membrane, there's another solution that is 10% salt. So it's 90% water and 10% salt. So which side has more water? The one with the less amount of salt. So when there's less solute, there's more water. So this side has more water. So if there's a membrane here that water can get through, but salt can't, if salt can get through too, it doesn't matter. They'll both reach equilibrium. The water will go from this side to that side until it reaches equilibrium. And that's what it's showing us here. So if we had, this is a special kind of a tube called a thistle tube, you don't need to know that. But on the bottom of the tube here is a membrane that water can get through, but the solute can't. So let's say it's sugar this time. So inside of the tube, we have 10% sugar. okay? And in the beaker, we have 5% sugar. So where is there more water? There's more water where there's less sugar. So that means there's more water in the beaker than there is in the tube. So water is going to enter, and the membrane, the sugar can't get through, only the water can. So the water is going to move into the tube until they can reach equilibrium. And if you look here, here's the level of water of solution in this side, and now it's up to here. And that's because water has entered the tube. And that movement of water from where there's more water to less water is called osmosis. And that happens a lot. It's important inside of our body because often what's inside and outside of cells can't move. Like you have big proteins and things, but water can move. So water will distribute itself across the cell membrane from where there's more water to where there's less water. So these are 3 definitions, and I'm going to go through the terms, but I'm going to use the picture on the next slide to do that rather than just read them off to you. So if you take a cell, so this is supposed to be one animal cell. We can say it's a human cell. This is a plant cell because plants are a little bit different. And if you take an animal cell and put it in a solution in which the concentration of water, oops, let me change it to, so if we put it in a solution where there's equal water inside of the cell and outside of the cell, that's called isotonic. So isotonic is equal inside and outside. So if it's already an equal amount of water inside and outside, there's not going to be any net movement of the water. So in an isotonic solution, there is no movement of water. It moves equally in and out of the cell. However, if you put a cell in what's called a hypotonic solution, so in a hypotonic solution, there's more water outside of the cell than inside of the cell. So there's more water outside than inside. So what does water do? It goes from where there's more to where there's less. So water will move into the cell. And it can continue moving in. If it doesn't reach equilibrium, our cells can actually burst open. And that's what it's showing here. And that solution is called hypotonic. So if you put a cell in hypotonic, there's more water outside of the cell than inside of the cell. So water will move in, and this looks like it's spitting something, but it's supposed to be exploding. The confusion, I think, that gets hard with this is that hypo you tend to think of as meaning less, but it doesn't mean less water. It means less solute. And so if there's less solute, there's more water. So there's more water outside than inside of the cell. And so water will move into the cell, and animal cells can actually burst. Suppose you did the other way, where you put a cell in a solution where there was more water inside of the cell than outside of the cell. In this case, this is called a hypertonic solution. And if there's more water in the cell than outside, then the water will leave the cell and go out into the solution. And the cell is going to actually shrivel up. And so hypertonic solution means not that there's more water, but there's more solute, so there's actually less water. So that is what's confusing, is the hyper part refers to the solute, not the water. The same thing happens in plants. On the bottom here, I know we don't talk much about plants, but you probably all have plants at home and stuff. And so if you put plant cells in hypotonic, they don't pop open like an animal cell will, because plant cells have a wall around them and it kind of prevents them from getting too big. And likewise, if they're hypertonic, they can shrivel up, but not quite as much because the wall is still there. So it's a little bit different in plants. So you're going to look at your, you have a passive transport lab, which you'll be doing, I think, next week. And you'll be making little bags like this with different solutes in. But this is just another example. So I wanted to go through it one more time because it is confusing. So if you have an isotonic solution, that means it's the same amount of water inside the cell. And so if this bag here represents your cell, it's 90%, 8% water, 2% sugar. And that's inside the cell, and then in the beaker, 98% water, 2% sugar. That's called isotonic. It's the same water, so you get no net change. However, if we put that same bag in a hypotonic solution, so in this case, the solution we're putting it in is 100% water. And remember, inside of the bag was 98% water. So that means there's more water outside than inside. So water is going to move into the bag. And you can see how the bag is swollen up in this picture. Suppose we put this bag into 90% water solution with 10% sugar. So now that solution is hypertonic, has less water, more sugar. So water is going to leave the bag and the bag is going to get smaller. The same thing would happen. These pictures here are red blood cells. Red blood cells are circles. They kind of are pinched in the middle. It's not really a hole like a donut, but if you put them in isotonic, they remain normal looking. If you put them in hypotonic, they kind of swell up. Some of them will even pop open. If you put them in hypertonic, they get all shriveled up looking like they do in this picture. So if you're going to get, for example, an IV at the hospital, it's important that that IV is isotonic. Because if it's not isotonic, that means your cells, especially your red blood cells, would either swell up or shrivel up. If you use, so suppose you didn't use isotonic, usually what we use is isotonic saline, which just is salt water. And it's the same concentration of water as inside of our cells, so our cells stay normal. If you were to use pure water for an IV, that would be hypotonic to your cells, and your cells would swell up and some of them could pop open. If you used hypertonic, that means there's less water. Your cells would shrivel up. An example of a hypertonic solution is ocean water. You probably know that you can't drink ocean water, and the reason is it's hypertonic. So it doesn't really quench your thirst. It dehydrates you even more because it's going to cause water to leave your cells if you drink it. So we mentioned when we were talking about, I thought I got rid of these, the types of proteins inside of a cell membranes, and that one of their functions of the proteins in cell membranes is that they enable things to get inside and out of your cell. And those carrier proteins are of two different types. Some are involved in what's called facilitated transport. So, facilitated, I can't say that today. Facilitated transport is still diffusion. It's still going from where there's more to where there's less. So it's still moving from high to low concentration. So it's still passive transport, does not require energy. And it's just using a carrier protein as a kind of hole to get through the membrane. For example, water gets into our cells. There's a carrier protein called aquaporin. So pore is like a hole, aqua is water, so it's like a hole for water to get through in our membranes that enables water to diffuse in and out of our cells. Sometimes you need things to be in or out of your cell that they're not moving from a high concentration to a low concentration, but moving from a low concentration to a high concentration. And so that's going against a concentration gradient. That does require energy, and it's called active transport. It's kind of like if your house gets flooded and all this water comes in and you want to get the water out, it doesn't diffuse out. You need to get a pump and plug it in to get energy to pump it out. So it's kind of that same idea that you're using energy. It also uses a protein in the membrane, but it couples that with energy use. So this is just showing facilitated transport where you have more of these little green molecules outside of the cell. They can't get through the lipid bilayer, but they use this carrier protein to be able to get in. And they'll keep moving in until they reach equilibrium and there's an equal number inside and out. The sodium pump, on the other hand, which is important in our nerve cells, is active transport. So it uses energy to pump sodium ions outside of cells in the brain. If you take anatomy and physiology, you'll learn a lot about how the sodium pumps work in brain cells to enable nerve transmission to occur. We're not going to go into that detail now, but that's an active transport system. So it requires energy to pump sodium out of your brain cells. So again, this is just a summary of the types. We haven't talked about bulk transport. We're going to talk about that next. I should have put this slide after that. So requires energy is active transport and bulk transport. We'll talk about the bulk transport in a minute. Does not require or passive transport is osmosis, which is the diffusion of water, diffusion of other things, and then facilitated diffusion through a carrier protein. The one thing we haven't talked about yet is the bulk transport. Just quickly talk about that. So sometimes, I thought I got rid of that. Sometimes you want things to go in and out of your cells in big pieces. And that's what they mean by bulk transport. We're not just talking about a single molecule. And that is a form of active transport. And it requires energy. And an example of this, this could be a white blood cell. So suppose if you go back to us having that strep infection. So let's say these little red things here are bacteria and you have an infection. And a bacteria can't get through the phospholipid bilayer or a carrier protein because it's a whole cell. It's too big. And so we have something called endocytosis. Cytosis is the cell process. Endo means to take in, where your membrane kind of folds around the bacteria and pinches it off and brings it inside of your cell, where it can then be broken down. So this is a bulk transport because it's a whole cell that's coming in. Likewise, sometimes like when you release insulin, like we talked about, into the blood, insulin is a protein. It's a big molecule. It can't get through the phospholipid bilayer or a carrier protein. So you package it into balls. We'll talk more about this later. And then you release a whole bunch of them outside. And that's called exocytosis. So exo means to go out of the cell. So endocytosis is when you bring bulk things into the cell. Exocytosis is when you do bulk movement out of the cell. And they're both active transport. They require energy. This is just a summary of all the different things. I'm not going to go over it now, but it's just for your own use. It kind of summarizes the types of transport across the cell membrane. So what we'll talk about next is the components inside of the cell and what their function is. So I'm going to have just one quiz for the part 1 and part 2 of the cell transport. Bye.